Okay, here we go for section 2 of this unit. This is only going to be a 2-slide video, and you're probably cheering now. It's probably the shortest video you've ever seen. That's because it's very hard to teach this on a video. So you'll see most of this in class on the board and with animations from on the video screen. But I'm going to give it a chance here. Remember, just write the stuff in green. So here's one big slide for you. Sorry about that. If you remember back when we did mitosis in the cell cycle, we had 4 phases of the cell cycle: G1, S, G2, and M. And I asked you to remember that the S phase is where DNA replicates. Well, that's this process. This is what we're going to explain. So instead of just saying DNA replicates, we're actually going to see how it does that. So DNA replication is the process of producing 2 identical copies of DNA from one original DNA molecule. So it's like taking one and turning it into 2. Okay, kind of like when your cells divide into 2 sister cells, or 2 daughter cells, sorry. It's a very complex process. I mean, extremely complex. In class, I'll show you a very complex animation that involves a lot of parts, a lot of enzymes, and then I'll show you the simple scaled down version that I want you to know. Basically, there's 4 steps. That double helix DNA can't be copied as long as it's in the helix. So there's an enzyme called helicase. Remember, enzymes usually end at ASE. That will unzip and unwind the DNA by breaking those very weak hydrogen bonds between base pairs. So C and G are together. It will pull C and G apart temporarily. Once those base pairs are broken, then they want to go back together, but there's going to be some proteins called single-stranded binding proteins. Don't write that down. Don't look for it on the screen. It's not there. Single-stranded binding proteins that actually hold them apart. So instead of having those bases go right back together, which wouldn't accomplish anything, any exposed bases will now pair up with new nucleotides that were formed in the cytoplasm. So they form in the cytoplasm, they enter the cell's nucleus, and once the C and G and the A's and T's have been pulled apart, new nucleotides come in complementary to those and base pair up, thus creating 2 brand new strands of DNA complementary of the original ones. And as I said, this is a little hard to even imagine. There's no way I can show it to you on the screen. So I just want you to get the words down now, and I'll show you better in class. Okay. Anyway, once you have this replication take place, you'll have 2 identical strands of DNA exactly like the original. Whoop, 3 minutes. That's not bad. See ya.